Hello, everyone. Good afternoon. First of all, I want to thank you, BRIC, for inviting me to present our recent work. And I also thanks my colleague, my students, and other professors who are supporting me for this work in Korea. Myself, Nagendra Kumar Kaushik, and I am working as professor in Kangwon University in Department of Electrical and Biological Physics. And also I'm working together as a participating researcher in Plasma Bioscience Research Center, also as a deputy director of PBRC. Today I will talk about one of our recent paper that is published in Journal of Bioactive Materials. Its impact factor was 17.3, and it is top 2% JCR rank in the world. And this topic is related to coronaviruses. As you know that we faced recently a very big pandemic, and we faced severe problems in healthcare settings. So we worked on it with this topic to treat coronavirus infections for future. And in this work, we used nitric oxide. So title of this presentation is Nitric Oxide Enriched Plasma Activated Water, which inactivates coronavirus 229E coronavirus and alter antiviral response genes in human lung host cells. So nowadays in PBRC, we are working on several topics. So some of those topics are mentioned here. So first, we are working on the virus inactivations, and for that purposes, we are using various kind of viruses such as coronaviruses, vaccinia virus, monkeypox virus, or various kind of pseudoviruses or influenza viruses for the treatment. And as well as we are working on waterborne, airborne, soil-borne pathogenic bacterial strains. We are also working on environmentally friendly approaches for the synthesis of nanoparticles for biomedical applications. And we are also working on various kind of plasma-based treatment strategies for green or smart agriculture methods. And finally, we are also working on some kind of cancers, which those are induced by environmental factors such as pollutions and other contaminations. And first, I want to tell you about plasma. What is plasma? You all may know about it. It is not blood plasma. It is a physical plasma, you can say. It's a fourth state of matter. Like solid, liquid, gas, we have three states of matter. We have further, if we give energy to gas, we can make plasma. That is fourth state of matter. And it consists of ions, electrons, reactive oxygen and nitrogen species, little amount of UV, very minute amount of UV, and some heats can be included in plasma. And this plasma can be tuned for various purposes. You can make your own plasma for generation of specific gas or specific reactive species by controlling physical factors or some kind of environmental factors. So this is about the plasma liquid interactions. Basically, when plasma is generated on the surface of device, it creates atomic species, mainly electrons, and some kind of OH, also some kind of very short-lived species such as superoxide and NO, and little amount of UV, as well as little amount of electric field. And those species, those kind of atomic species and these short-lived species are called primary reactive species, which can further react with the nitrogen and air, oxygen present in the ambient air, or water present in the ambient air to make more stable species. So those are secondary reactive species. These species can be NO2, NO3, NO, and HO2, singlet oxygen, S2O2, ozone. And further, these species react with the target. Target can be water, target can be the human body, target can be the patient organ, a target can be plant or bacteria or viruses. On those targets, they further react in the presence of water. They further react and make more stable species. Those are tertiary reactive species. So every species have their specific role. So you can control these species in plasma by controlling physical factors. So these are some devices developed by PBRC. This is one kind of soft jet plasma. You can see here the soft jet lume. This is microwave jet plasma. This is FEDBD jet plasma. You can see this plasma lume. And this is FEDBD, floating electrode dielectric barrier discharge plasma. And their physical properties are mentioned in the yellow color on these pictures. And this is micro DBD plasma. It have several more than 100 micro cells. So that's why we say micrometer cells. That's why we say it micro DBD plasma. Its size is 90 millimeter and more bigger, same kind of device, bigger size, up to 30 centimeter we can make in PBRC for the large surface area treatment. And this is also micro DBD plasma. This is annular jet plasma, and this is one kind of thundering mimic device. We called it as nanosecond pulse plasma. So these devices can be used for specific application in biomedical areas as well as environmental areas, agriculture, or microbiology. So this is the video. You can see this is Plasma Go. This is Plasma Aurora. This is Plasma Jet. You can see plume here, this area. This is micro DBD plasma in live state. And this is another big size micro DBD plasma. This is another floating plasma jet. This is floating plasma type. You can see plasma streams, several plasma streams in this plasma. This is a cylindrical type plasma. You can see plasma generation here. And this is plasma inside water, plasma generation inside water. You can see plasma generated plasma here. And this is live state of nanomaterial synthesis by using jet plasma. This is gold nanoparticle, live synthesis by using plasma. And this is seed treatments for agriculture purposes by using plasma devices. And this is the device for ambulance air filter, air sterilization. So this device is very useful during the MERS time. It is installed in more than 5,000 ambulances to inactivate MERS virus in Korea in 2016-17. So these all plasma devices can be used for specific applications. Nowadays, we are focused on agriculture as well as environmental applications, as well as cancer treatment, stem cell differentiation, stem cell activation, or immune cell activation, or you can say immunomodulations. So several applications we are doing at Plasma Bioscience Research Center at Kangon University. And our main focus after Corona pandemic was viruses, how we can inactivate viruses by using those kind of non-thermal plasma. So in this slide, I am showing you the comparison of non-thermal plasma with radiations or radiation therapy, and a comparison with photodynamic therapy. So we compared all these three things: non-thermal plasma, radiation therapy, and photodynamic therapy. You can see here non-thermal plasma properties, radiation treatment properties, and photodynamic therapy, or you can say PDT properties. So mainly we compared many things, but two things here were major effect. One is side effect, and one is direct effect on the immune cells. So side effect in radiation therapy is already known. It is very dangerous. We can have very dangerous side effects. Skin can changes, rashes can occur, or second cancer can occur at another body organ, and it can damage to the nearby body organs also. And DNA breakage is the big problem during the radiation therapy. And in PDT, even PDT is the safest one, but still we have some kind of skin changes problem in PDT therapy. But in case of plasma, any side effect is unknown. There is no major side effect at moderate doses of plasma to the human being or animal or cells. Second main major effect was direct effect on the immune cells. Immune cells are very important part of our body defense. So we have to check whether these kind of therapies are have negative impression or positive impression on immune cells or immune system. So we checked all these three things and we found radiation therapy have suppressive in nature for immune cells. It used to suppress immune cells. Even PDT is the safest therapy and very recent therapy used to treat cancers. That is also suppressive in nature. So it is the drawback even in PDT. But however, in case of non-thermal plasma, it shows preservatory effects or stimulatory effect. Means it have good effect, positive effect on the immune cells. Either it preserve those immune cells, don't show any effect on this immune cells, or either it activate immune cell to fight against those kind of diseases or cancer. It can be cancer, it can be infection. So these are two main things which can help us to bring plasma in hospital settings or clinical settings. So these are previously I showed you all basic things. Now I will start about the inactivation of viruses using non-thermal plasma. Non-thermal plasma for inactivation of viruses is not new thing. Already people did on bacteriophages or many other kind of viruses to inactivate them. So various type of viruses, morphological, they can be morphologically different, are treated by non-thermal plasma previously. And these viruses are inactivated based on reactive oxygen species, reactive nitrogen species, UV radiation, ions and electron present in the plasma. And basically plasma can target viral nucleic acid present in these viruses and protein of these viruses. So it can have effect by targeting nucleic acid or damaging DNA, RNA, whatever virus is that, or proteins like surface proteins or membrane proteins and other proteins. So the mechanism is very simple. Plasma target DNA, RNA and degrade it and degrade its proteins and make them ineffective particles. So these ineffective particles after treatment by plasma cannot infect human or animals again. So these are just inactive, damaged virus particles. They cannot infect anyone after this treatment. So this is already reported things. But during Corona pandemic in 2020, it started in March in Korea, as I remember. We thought we can try to check inactivation effect of plasma on viruses. So we tried many strategies and we focused on the three main things. One is virulence factor of virus. That is in SARS-CoV-2 case, it was S protein on the virus. And one is virus binding and entry. Can we block virus binding and entry to the human host cells? That was our second question. And third important topic was host cell response. Can we control like host or human patients immune system, or can we control those kind of immunomodulation during virus infections, or can we control blockage of blood vessels or blockage of heart or many other side effects during the virus infection by using plasma? So these were our three topics, and we are nowadays also we are working on these three topics. We are targeting virulence factor by oxidation or attenuation by plasma, or we are making them functional impaired. So they cannot infect host cells easily. Second thing, we are also checking the how we can stop these viruses to go to the human lung cells or human host cells. And third, we are working on immunomodulation effect by plasma during virus infections. And lung rejuvenation, can we rejuvenate those human host cells which are infected by the viruses or not? And we are also working on the can we create vasodilation, because many people died in coronavirus pandemic due to the blockage of blood vessels. So vasodilation was necessary that time, but drugs are not very effective. So we are also working on these three topics if we can control it, then we can control viral pathogenicity, and we can control even more dangerous viruses in future by using plasma strategies. So basically, in plasma, we can generate nitric oxide species, hydroxyl species, and this is singlet oxygen, S2O2, ozone, and some active active species, and little amount of UV. In this case, we used to thinking before that these all species are important for the virus inactivation. But some of these species can have toxicity. So we chosen NO species are more like it is biosignaling molecule in the human body. So we can use this NO species for virus inactivation easily, because it is already in our human body. It is present already in our human body at very minute concentration. And our body also work on that NO species level. Many signals are based on the NO species level inside our body. So we targeted this species by the plasma. And now I will tell you some other studies we did on coronaviruses. Like this is the one study by the plasma community that they inactivated viruses on the surfaces, because the infection was always through the surfaces or touch or by the direct exposure by the infected patients. So we checked plastic, metal, cardboard, football, basketball, and baseball. And these surfaces are infected by the coronaviruses, that is SARS-CoV-2. And after that, those viruses present on these surfaces are treated by the jet plasma, this kind of jet plasma. And later we checked the effect and found significant inactivation of viruses happen on this all 6 surfaces. So you can see this graph, this red line showed after treatment and blue line is before treatment. So significant reduction occur on all surfaces. We found very slight effect on football, but otherwise on cardboard, it had big effect, baseball, as well as this plastics, basketball, and metals. So it shows that we can sterilize surfaces by using plasma. Those are infected by the viruses. In another study, it is shown that can we damage, this is the virus particle, SARS-CoV-2 virus particle, as you remember. It have this S protein. So we thought, can we damage this S protein? And on this S protein, there is a specific domain that is RBD, receptor binding domain. So can we damage this receptor binding domain by plasma or plasma treated water? For this purpose, we prepared plasma treated water. We used this micro DVD plasma, micro DVD plasma. We generated plasma on the top of this water, and many species generated in this water are solubilized. And then we treated this virus by this plasma treated water and found the RBD domain is damaged, and interaction between RBD domain and ACE protein. ACE protein is present on the human lung host cells. So this RBD of virus and the ACE2 of the human host cell must interact. This interaction is gone after this treatment. And also we found that this after damaging this RBD of virus, these viruses cannot enter to the human host cells, so cannot infect. So the virus titer was reduced a lot in this study, even by this plasma treated water. Further, we planned to make a very specific plasma treated water in which we can have just NO, because NO is very safe for human being. At some concentration, we can treat human being by NO species. So NO is a very nice biosignaling molecule inside human body. It controls heart, blood vessels, even nervous system in many ways. So in this process, we used microwave based plasma. So microwave is generated by the magnetron, and it goes through the circulators and then goes through the 3 subtuners. And this microwave hits in this quad chamber to produce very high temperature plasma. And this plasma is generated in presence of 2 gases, nitrogen and oxygen, only 2 gases, nitrogen and oxygen, to make pure nitric oxide. And in this high temperature, pure gaseous nitric oxide is formed. which is cooled down before exposure to the water by the water cooler. And we got very high concentration of nitric oxide water after this gaseous NO exposure to the water. And this is the process. It makes atomic nitrogen and react with the oxygen, and finally it makes gaseous NO. And this gaseous NO is imposed on the water to make nitric oxide plasma activated water. This nitric oxide plasma activated water further used to inactivate virus, and we used here human coronavirus 229E strain. We standardized this preparation of nitric oxide water by changing many physical parameters such as amount of water, nitrogen gas, oxygen gas, even power. Power is almost same, but the exposure time is changed. So by changing some parameter, finally we got 4,000 micromole of nitric oxide plasma activated water concentration. So concentration of NO in this condition, 3 condition, was 4,000 micromole, and pH was also not very low. It is around 4.3 to 5 pH, which can be used further by dilution in the clinical settings or to the patients or to the animals. Later we checked physiochemical diagnostic, and by gas FTIR, this is the live gas FTIR data. And by gas FTIR, we found that mainly this plasma produces nitric oxide, not any other species, not N2O, very little amount of NO2, that was also minor, no CO2, no other species. So you can see graph here. The concentration of NO was maximum, up to more than 4,000 ppm. However, the concentration of NO2 was very less than 75, and N2O in points, nothing, it's negligible concentration. And other species like HNO3 or ozone are totally absent. Their level is just 0. So it means we are producing just nitric oxide by using this plasma. And this nitric oxide further goes to the water and forms nitric oxide species. And we check the nitric oxide species in the liquid. This is the gas phase data. This is liquid phase data. We checked in liquid phase nitric oxide concentration, and we found 4,000 micromole concentration of nitric oxide inside water. Further, we checked the stability, aging, or you can say decay of this nitric oxide, because by time it may change. So for 14 days, we studied at 3 different temperatures. One is room temperature. Room temperature was 25 to 30 degree during that time. And we checked on the 4 degree in the Ninjangu, or you can say fridge. And also we checked on the -80 degree. We found if we store this nitric oxide water in -80 degree, we can retain its concentration for a very long time, even for years. We personally tested in our laboratory. Even for years, the nitric oxide concentration can be maintained in -80 degree. But in 4 degree, few weeks is okay. After that, it will reduce. But in room temperature, few days are okay. And after a few days, it will reduce. So this is the aging data of this nitric oxide water. And we also checked S2O2 and other species like OH in our study. And we found the S2O2 and OH, both are negligible in our nitric oxide water. And we also checked conductivity and ORP. Both are increased at condition 3, which is the conductivity showed around 1300 microsecond per centimeter. And also ORP is increased at condition 3. This nitric oxide water further we used to treat coronaviruses. So first we checked cytopathic effect. So cytopathic effect, this is control, this is placebo, just water, no plasma. And this is plasma treated water, NO, nitric oxide plasma activated water at 150 micromole concentration, 300 micromole concentration, 500 micromole concentration, and 1000 micromole concentration. So we tried various concentration of NO water and found the cytopathic effect by the virus. Virus have property to infect human cells and make the cytopathic effect. So we checked whether this cytopathic effect is reduced after this plasma treatment or not. And we found 2 log order difference up to 1000 micromole of nitric oxide water. So you can see this graph. This is easy to understand. This is control, and this is 1000 micromole NO water. So 2 log order changed. The cytopathic effect is changed by 2 log order. It is reduced basically by 2 log order by this treatment. Even 300 micromole and 500 micromole showed very significant activity. So it is very good news for us that we can use this nitric oxide plasma activated water even for the treatment of the patients or animals against virus infections. Further, we check the therapeutic effect, whether we can use this NO water for therapy in human patient or not. For that, according to medical standards, we need to check therapeutic index, that is SA index, or you can say selectivity index. For calculating selectivity index, we have to measure cytotoxic concentration 50 and effective concentration 50. So cytotoxicity concentration 50 is calculated on human host cells. Whether this agent is cytotoxic or not, this is cytotoxic concentration 50. And effective concentration 50, that we say EC50, is based on the virus inactivation. So whether this agent can reduce viruses or not. So we calculated CC50 of our NO water and nitric oxide plasma activated water, and we compared this effect with market available 2 drugs, chloroquine, hydroxychloroquine, and ritonavir. This is well-known drug during the pandemic. This is also well-known AIDS drug, but it is also used to treat coronaviruses. So these all are refurbished drugs. And we found that the cytotoxicity by this NO water is very less, and it shows only cytotoxicity after 2500 micromole concentration. That is very high. So it means this is non-toxic below 2000 micromole concentration. So we can use this any concentration below 2000 micromole of NO-PAW. In case of chloroquine, highly toxic. Even at after 100 micromole, it is showing very significant toxicity to the human cells. So we cannot use it at high concentration. It can have some kind of side effects in the human patient. In case of ritonavir, another AIDS drug which is used for coronavirus treatment also, it is also toxic after 200 micromole concentration. These both drugs are toxic, can have some kind of side effect to the human patients. And we checked whether these 3 agents can kill virus or can effectively reduce virus. So we found that yes, NO-PAW can reduce viruses based on the concentration of NO-PAW. Same in case of chloroquine, it can also reduce viruses, and ritonavir also, it can also reduce viruses after 150 micromole of concentration. So we calculated by using these two data, CC50 and EC50, we calculated SI index. In NO-PAW case, CC50 was 2402 and EC50 was, the effect on virus was 70.8 micromole. And SI index we calculated by dividing CC50 by EC50, that is 33.8. In market available drug, you can see SI index is 11.5, very less than the plasma activated water, NO water. Same case in ritonavir case, 6.2, more lesser. SI index or therapeutic index or the therapeutic values of these two market available drug is far less than nitric oxide plasma activated water. So it is a very good news that we can have wonderful therapeutic strategy to treat viruses in future by using nitric oxide plasma activated water. We also checked not only therapeutic value, we checked the gene expression after treatment by nitric oxide plasma activated water infected human host cells. In this case, we specifically checked two kind of sets. One set is for viral genes, viral loads, such as N gene, virus E gene, and virus-related M genes. So 3 genes we checked. We found these 3 genes of viruses infected host cells are reducing after this treatment by nitric oxide plasma activated water. So you can see the concentration dependent decrease in these genes, virus genes in the human host cells. In all cases, all three cases, it is reduced. And this effect is more better than the market available drug. These are market available drugs. So the effect is more better. Inhibition effect is more better than the market available drugs in all three cases. And we also checked another set which is related to human host cells, human lung host cells genes. And these genes are responsible for antiviral response to fight against virus. And in this case, we found these genes are increasing dose dependently in many cases, data showed significant effect in many gene cases like BIRC3, DDIT4, FOSL1, or interferon related factors such as IRF1. Those genes are increasing. So it means not only this treatment is reducing virus, this treatment is also making your host cells, your lung cells, or your body cells more stronger to fight against viruses. So we can have a dual effect by one solve, just one treatment. So this is very hopeful study for future therapy in patients or in animals. And we also checked another molecule that is CD13. This is the receptor present on the human host cells. And this receptor is responsible for the entry of coronavirus in the human host cells. And we found there is no such effect. There is no significant effect by this treatment on the human host cell receptor. It means this treatment is just controlling virus and controlling immune defense system of cells, but not affecting the receptor by which virus goes to the human body. And this is important also because this receptor is also responsible for many biological activity, not only virus entry. This is important for many other biological activity or many other biological signaling. So we cannot damage those kind of receptor just to protect from virus infections. And further, we check the S protein also. You can see these pictures. This is control, and blue color is nucleus, DAPI stain, and this red color is virus S protein. So you can see this is virus control in human host cells, and this is treatment. This whole red color, the virus spike protein or virus S protein is gone after this just 100 micromole treatment of NO-PAW. And by the market available drug, you can see little red color still exists. That means this NO-PAW is more effective than this market available hydroxychloroquine drug, which is used for the virus inactivation. So this is graphical representation. You can see effect here. NO-PAW have reduced virus S protein more as compared to control, or as well as compared to market available drug, that is hydroxychloroquine. So NO-PAW have very nice effect, very significant effect on the virus S protein reduction or inactivation. And we also check this data by the flow cytometer. So you can see here, this is virus S protein control, and this is treatment by 100 micromole dose of NO-PAW. So it is reduced. This fluorescence shows this S protein flow. This is at x-axis, you can see S protein fluorescence intensity. This intensity is decreasing after treatment with NO-PAW, and this is graphical representation of this flow cytometer data. You can see as compared to virus control, NO-PAW is reducing virus S protein significantly, very significantly. And finally, we reported the mechanism in this paper. Not only virus reduction, it is also damaging the S protein of the virus, which is responsible for the less entry to the host cells. And even if they can enter to the host cells, then our treatment strategy is reducing viral genes such as viral N gene, viral M gene, and viral E gene to reduce further proliferation. And also, not only this, it is not only affecting virus, it is also affecting the human host cells to express high amount of antiviral response genes which are responsible for the defense against virus infections. So we can have many things by this treatment, 3 things by this treatment. And finally, we can have very nice antiviral strategy for inactivation of coronavirus. That is reported in this paper. Further, in the extension of our work, we also worked on plasma-generated ozone, not for treatment, not for human treatment or a patient treatment. It is just for sterilization of surfaces. In this case, we generated plasma ozone, and we treated these viruses in our lab by this plasma-generated ozone, and we found very nice effect. You can see EC50 virus effect on viruses by this treatment is very high, and EC50 lies between 6 or 7 ppm of ozone. And this is the CC50 data, and this is not toxic up to 10 ppm. So we can use even up to 10 ppm, we can use in room to sterilize tables or surfaces, plastics or doors, or like surfaces, other surfaces like computers or keyboards, those kind of things. We can, even in hospital setting, we can use this method to inactivate viruses on beds, patient beds, or patient settings, or hospital rooms, or in emergency rooms. So this strategy is also working, and we can use this only for sterilization of virus on the surfaces. We cannot use this strategy for the treatment of patient, because nobody will like ozone. No human will like ozone to be as a medical agent or clinical agent. So it can have some side effects. So we don't recommend this plasma-generated ozone for the real treatment process. Further, we are also, in extension of our previous work, we are also working on the virus inactivation synergy by using metabolic modifiers. And for this purpose, we are using 2-DG, 2-deoxy-D-glucose molecule, which is making host cells less metabolic active. So the virus cannot be propagated in the host cells. So this strategy is also working. We are working on with plasma and 2-DG together to make virus ineffectivity very like more than 3 or 4 log order. So this graph, I will not tell you the whole data because it's complex data. You can see this graph, color graph. We got up to 4 log order difference as compared to control. So we reduced virus by 4 log order by this combination treatment strategy. Almost virus is gone after this treatment. So this strategy is also very beneficial and very nice input in the medical settings, that if we have some serious patient, we can use 2-DG to control metabolic activity of lung host cells. So virus cannot propagate easily in those lung host cells. And that at the same time, we can treat those infected cells by the plasma to kill leftover viruses. And nowadays, we are also working on the many kind of mutated strains of SARS-CoV-2, and results we will report soon by plasma and plasma activated water. So we are using 2 kind of strategies here, cold plasma as well as plasma treated liquids or water. And we are also working on some strains related to monkeypox viruses. In these cases, we are focused on entry fusion complexes. So these are the 12 proteins which are responsible for the entry of monkeypox virus in the human host cells. So we are focused on whether we can damage these proteins by the plasma or not. So that is our future study. We will report soon. And we are also working on the whether this nitric oxide plasma activated water can inactivate various kind of bacteria present in environment or not. For that purposes, we started E. coli or P. aeruginosa and S. aureus bacteria for inactivation effect. And we got very nice result that is reported in International Journal of Molecular Science recently. So we can inactivate it, even environmentally transmitted bacteria, by using this nitric oxide plasma activated water. So this is same study. The more data, we can damage the surfaces. By using this strategy, we can also activate nitrosative and oxidative stress genes to kill bacteria, and we can alter the homeostasis related genes for controlling the bacterial metabolic activities. And not only NO water, we are also working on the plasma treated liquids for that for various kind of biological application, such as cancer treatment or bacterial treatment, environmentally transmitted microbes, or virus treatments. The same kind of effect we got here. We can have more than 5 or 6 log order difference by this treatment. So we are reducing bacteria by using just plasma treated liquids. So conclusion of this study is that NO-enriched plasma activated water, that is NO-PAW in the study, was prepared using microwave plasma generated gaseous nitric oxide. And this NO-PAW inactivated human coronavirus 229E strain. And NO-PAW showed no significant toxicity below 2000 micromole concentration. So we can use any concentration below this 2000 micromole for human patients or animals or any other subject. NO-PAW reduced viral RNA load as well as spike protein in the infected host cells. Also, NO-PAW elevated antiviral response genes in the infected host cell to fight against virus infection. Also, plasma generated ozone, nowadays we are working, can inactivate virus on the surface. That is not for therapeutic purpose, that is just for sterilization purpose. And NO-PAW is also effective against environmentally transmitted bacteria. And we can say it is a very nice sterilization agent, can be used in hospital settings or clinical settings. Also, NO-PAW can be used for smart and sustainable agriculture processes. That is not related to this paper, but we are working on it. We may report this work soon in high impact journal. This is our one device collaborating company, that is PlasQ Air, based on the plasma inactivation of coronaviruses. And we as well as the certification agency by the government of Korea found that 99.9% coronavirus inactivation can occur if we have this machine in the 33 pyong of house or maybe more bigger size rooms. Not only coronavirus, this machine also inactivates superbacteria up to 99.9%. So recently our company got many awards for this work, and more than 5,000 devices are sold out in China, Singapore, or neighboring country, as well as in Korea. So this is real device in my home. So fish are enjoying. This is fish tank. Fish are enjoying every time. There is no fungus, no algal development in our fish tank in presence of this PlasQ Air device. This is another device by our company. This is our company PlasAid. And this PlasAid device is for agriculture purposes. So this device we are proposing for green and smart agriculture. It can have 3 kinds of effect. This plasma device can sterilize air and water near the water irrigation system. So bacteria, fungus, and viruses can be controlled in the water which is used for irrigation. Second, this device can be helpful for controlling bacteria, viruses, and fungi in soil and plants. And also, very important thing, this device can act as a fertilizer. It can produce plasma activated water. That water can act as fertilizer for the nitrification in soil for plant growth. So this is very nice technology. We will more focus in future this device for development of smart agriculture system. And finally, I thank BRIC. I thank all post-tech members who invite me for this nice seminar. And this is our director, department head, Professor Yunha Choi. And these are my PhD students. And now this student is postdoc in USA. He worked with me also postdoc as a postdoc. And these are all members, collaborating members who are working with me for any kind of research related to plasma or nanomaterials. I thank all my PBRC center members and department members, our colleague professors for helping me in this work. And this is my address and email ID. If anyone have any question or comment or need any help from our side, please contact us on this email ID. Thank you very much. And thank you all audience for listening carefully and patiently this presentation. Thank you.